Welcome to Introduction to Ecology, Lesson 22. This is a slide we've seen a lot before, probably at least 5 or 6 times, and it's really that organization of ecology. And so far in this course, we've been sort of focused at the population level. And then we scaled up a little bit and we looked at maybe pairs of interacting populations. And then in the last 2 lectures, we finally got a little bit more complex and started to look at communities. Well, now we're going to take a big leap and move to landscapes and biospheres. Basically that large scale of ecological study where we're looking at things happening across very broad regions or even the entire globe. That leads us to the study of biogeography. And I'm going to give you a little bit of a story about my life and how you can see the effects of biogeography in my life. So I'm a Canadian. And in Canada, we celebrate Thanksgiving in October. And every Thanksgiving, my family takes a hike up this rock sort of cliff, and we just look at the fall colors. But one year, a couple years ago, as we were on our walk, we ran into this guy. This is a Massasauga rattler. And Massasauga rattlers are native to Ontario. But for many years, since long before I was born, Massasauga rattlers have been very rare. And so most of us have never seen a rattler in Ontario. And so when we saw the Massasauga rattler, we had a particular response. This is my cousins with the rattlesnake, and you can see my one cousin there posing and making faces, and you can see her small child, probably about 7 or 8. And she's not super concerned about her small child being really into this snake and getting close to it. And this is my sister-in-law. My sister-in-law is not from Canada. She's actually from Georgia. And turns out in southern Georgia, where she's from, rattlers are not that uncommon. In fact, rattlers are pretty common. She's seen lots of rattlers in her life. And so as opposed to our response to the snake, this was her response to us. She just didn't get it. She could not believe how we were responding to this venomous rattlesnake on the rock. And really, there might be different reasons. Maybe my family loves animals. Maybe Canadians love animals. But really, I don't think that's the case. I think it comes down to biogeography. And the fact is that we never see rattlers. So when we see a rattlesnake, we get really excited. It's a cool thing. It's just so unusual. And we take pictures by it. Whereas where she comes from, rattlesnakes are pretty common and nobody gets excited about them. In fact, they're so common, a lot of people are afraid of them. And so that's really a pattern that we see, and that's sort of one of those patterns that we're interested in studying when we study biogeography. So biogeography is the study of the variation in species composition and diversity among geographic locations. Very broadly, species richness varies from one location to another. And you can look at this variation across different groups of organisms, different types of plants, different types of animals, anything really, different types of bacteria even. And what you'll notice is that, there are certain regions where you have a lot of diversity and a lot of abundance of particular organisms, and there are other regions where you have very few of those organisms. This is just one example, and this is actually salamanders. And it turns out that, sometimes we think of these cool places like the tropics having amazing species diversity, and a lot of ecologists want to go to the tropics to study species diversity. But in our own backyard, in the southern Appalachians, right where we live here in Clemson, we have the highest richness of salamander diversity. That's sort of something special about our region relative to anywhere else in the globe. And so there's a variety of different species of salamanders that you can find within 4 or 5 hours of Clemson, even within Clemson. And the neat thing is, because we have such high diversity, we also have what we call endemic salamanders. In fact, many of the salamanders that live in the southern Appalachians are endemic to the southern Appalachians. An endemic means that they occur nowhere else on Earth. The only place they're found is in our backyard. Now, salamanders are a little unusual. They're sort of different patterns that you see in different groups of organisms. So here I've got 3. I show you amphibian diversity, I show you mammal diversity, and bird diversity. And you can see that those don't quite look like salamanders. We don't have that little red spot right along the ridge of the southern Appalachians. Instead, we tend to have most of the diversity in these 3 groups of organisms centered closer to the equator. And so one really common trend in species richness is that it varies with latitude. And by and large, for most types of organisms, again, salamanders are an exception, but for most types of organisms, lower latitudes have many more different species than higher latitudes. So they just have sort of more diversity and even more abundance. But latitude doesn't explain everything. So we actually find that even at the same latitude, species richness and species diversity and species composition varies from one continent to another. So even if you look at along the equator, here for example we've got bats, and you'll see that there's a lot of diversity of bats near the equator, but only in South America. You don't see that same diversity in Africa. And in fact, in South America, there's a lot of really cool types of bats. There are actually bats that fish. Frequently, we think of bats as being insectivorous, and so they eat insects, but there's actually bats in South America that fish, and there's bats that eat blood. Vampire bats. They actually do exist. They're a real thing in South America. Now, fun fact, the story of Dracula by Bram Stoker was not from South America. In fact, when he wrote Dracula, they didn't even know about vampire bats. It was sort of something he invented. He was from Europe. But they do exist, and there's a lot of different sort of feeding and lifestyles of bats in South America, but you don't find that as much at sort of low elevations along the equator across the other continents. Another interesting thing about biogeographic patterns is that the same type of community, the same type of biome can actually vary in species richness and composition depending on where it is. So way back in one of the early lectures, we found that evergreen forests are located along the coast of the Pacific coast of North America and also in New Zealand. And actually, if you look back to that lecture, what you'll find is that these forests, I even mentioned it there, these evergreen forests are much more diverse in New Zealand than they are in North America. So same type of biome, they're classed as the same plant form, and so that, remember, the plant form defines the biome, but within those forests you have very different amounts of species richness and species diversity. When we talk about patterns in biodiversity, we have to watch because depending on the scale we look at it, we get different answers. So if we look at broad scales across the entire globe, sometimes the way we interpret patterns in biodiversity can be different than if we look at smaller scales, maybe across North America or across the Southern Appalachians or even in our own backyard. So the largest scale is what we call the global scale. And when we're looking at patterns in species diversity at the global scale, we have to keep in mind that these species have been isolated by long distances, so continents, oceans, over long periods of time. And so these long-scale evolutionary processes are really important to describing the differences and patterns that we see in biodiversity across that kind of scale, across those really large scales. And here's an example where we have placental mammals and Australian marsupials. And you can see that basically the marsupials have filled a pile of niches that are also filled by placental mammals in other places. But we have that sort of very different species composition in the two places, even though the niches are the same, and that's because of those long separations over long evolutionary timescales where we had primarily marsupials filling all the niches down in Australia, and we had primarily placental mammals filling all the niches in North America and in Europe. So the largest scale is the global scale. If we move down a little bit, we get to what we call the regional scale. So these are smaller geographic areas where climate is about the same and where species are kind of restricted by dispersal limitation. So this is just a graph of the ecoregions in the Southern Appalachians. And so you can see that depending on where you are, both in terms of latitude, but also in terms of along the sort of elevation of the Smokies or of the Southern Appalachians, you have different ecoregions. They have different climates, and they have different sort of precipitation patterns and temperatures and that sort of thing. And so again, different species are found in these different ecoregions. And because those ecoregions are not found anywhere else nearby, those species are primarily stuck in those ecoregions. Now within the ecoregions, the physical geography that's the landscape, what we call the landscape, is very important to determining what's going on within that region. And so a good example is this Pigeon Mountain salamander. So this Pigeon Mountain salamander is found in that little top corner of Georgia, the northwestern Georgia. And what you'll notice is that that's sort of in that yellow ecoregion. But the salamander is not located across the entire ecoregion. That's the Ridge and Valley ecoregion of the Southern Appalachians. Instead, it's localized in that one little gray patch. That's the only place on Earth where it's found. And the reason that it's localized in that one little area and not spread throughout the entire ecoregion is that it's restricted to a particular physical feature of the landscape. It's restricted to a cave system, and that's where it evolved, and that's where it's found, and that's the only place it's found. And it doesn't move out through the rest of the ecoregion because of this very specialized physical environment that it needs. So we move down a little bit smaller. So again, global scale was the biggest, then we moved to regional scale. We've got all the same climate, and it's sort of a localized little area where it's hard to find another climatic region that's similar, but within that we've got sort of this broad area that's sort of shared. If we move down even further, we can get to the local scale. So we might have the Ridge and Valley ecoregion of the Southern Appalachians, but within that ecoregion there's sort of variation in the habitat that's available, and that leads us to the local scale. So the local scale is basically equivalent to a community. And you can think about hiking if you've ever hiked in the Southern Appalachians, and it's not all the same. As I'm hiking along, even at the same elevation, I'll come to a little patch of clearing in the forest, and then I'll come to a little patch of deciduous trees, and then a little patch of maybe some conifers. And so you have this variation at the local scale, and within that local scale, that's where your communities are found. And so here's a good example where, I might be along a mountain in the Southern Appalachians, and in one area I don't have a lot of streams. It's much more dry and a bald, and so I'm not expecting to find salamanders there as much. Whereas in another region, just a couple meters away, maybe I have a river going through, and so that's where I expect to find the salamanders. And so again, that's that local scale variation. It's all within a single ecoregion, but there's that variation from one place to another depending on where the rivers are and where it drains, and sort of where you have a little indent that's protected from the wind or snow. And so again, these local scale patterns and diversity reflect the suitability of biotic and abiotic characteristics of the habitat on sort of very small scale patch levels within that broader ecoregion. Now, to measure diversity at all these scales, whether it's at the global scale, at the regional scale, or at the local scale, we have three different sort of types of measures. So far we've talked about Shannon diversity, which is a quantitative metric for just putting a number on the amount and evenness of the different species that you find in an area. But now we're going to look at trying to sort of compare diversity or measure diversity on different scales and across scales, and for that we have these three additional measures. And so the first one is gamma diversity, and this is a measure of all the species within a region. So here you can't read it, but don't worry about that. This is a list of all the salamanders that have been found in the Southern Appalachians. And so this reflects our gamma diversity. The gamma diversity is also referred to as the regional species pool. It's all the species that live in that entire sort of ecoregion. So here, the ecoregion would be the Appalachians in general, and all the salamander species that live there are in this list, and that's basically my regional species pool, and that's what I use to figure out my gamma diversity. Alpha diversity is a measure of diversity at the local scale, at a very small scale. So I have my salamander checklist for all of the Southern Appalachians, and that's my gamma diversity for the region. And then within that, if I look at one single mountain, maybe I don't find all those species. Maybe I find this particular species and maybe one other and another and another. And those are the only species I find on that particular mountain. Well, then the sort of species I find on that mountain, that's the alpha diversity for that mountain at that local scale, at that one single mountain within the Southern Appalachian ecoregion. The final measure of diversity is beta diversity. And this is the change in species diversity or composition from one community to another, from one local scale region to another local scale area across the landscape. So here I've got my one mountain, and let's say this is a mountain in Asheville, and I've got my 4 different species, and that's my sort of alpha diversity on that mountain. Then if I take a look at another mountain, maybe one over in Hendersonville, and I've got the same 4 species, that's my alpha diversity for Hendersonville, if they're exactly the same between Asheville and Hendersonville, I would say I have low beta diversity. I don't have much change in the species in going from the local region in Asheville to the sort of local habitat in Hendersonville. On the other hand, if I take that same mountain in Asheville with those same 4 species, and then I look at Hendersonville and I still have the same alpha diversity, I've still only got 4 species there, but all of a sudden they're different species than what I have in Asheville, I would say that's high beta diversity. I have a high turnover, a high change in the species present going from one local area to another local area nearby. That they all have to be sort of in the same landscape and sort of the same ecoregion. All right, sometimes we are interested in understanding how local and regional species richness is related. And to do that, we tend to plot local richness as a function of regional richness. And this can actually tell us not just sort of how the two are related, but a little bit about the processes that drive local species richness. So the sort of theoretical upper bound when I'm plotting local richness as a function of regional richness is a straight line with a slope of 1. In this case, the local richness equals the regional richness. So basically, everything you find at that local scale on that one single mountain, you also find in the entire region. And that has to be the theoretical upper bound because to find the regional richness, we add up sort of the number of species in all the different local patches. So there's no way that I can have more species in one local patch than I have in the entire area that includes all the local patches. So again, when I have a straight line equaling 1, that's when local richness equals regional richness, and everything that's found over the entire region is also found in every single local patch. Typically, that's not the case. Typically, I have one of 2 other options. So I might have a straight line with a slope less than 1. So in this case, local richness is proportional to regional richness. So I don't have every species that's found in the entire region is not found in the local area, but when I have more species in the region, I have more species in each local area. So here again, that's the line in blue, and you can see that again, if I double the number of species in the large region, I'm going to double the number of species on average in each local habitat, but I don't necessarily have the same number of species in the whole region as I have in each local habitat. The final option is where the curve actually levels off. It's not a straight line. So in this case, the local richness stays the same as the regional richness gets bigger and bigger. And that's that green line right there. So in this case, again, for a little while, my local richness increases as my regional richness increases, but then at some point it kind of stops increasing. And even though my region may have more and more and more species, each local patch only has the same number. So when I have a straight line with a slope less than 1, that's an indication that regional processes are dominating. Regional processes are determining the species richness of each little local habitat. However, when I have a curve that levels off, right, that's an indication that local processes are dominating, that local processes are determining the species richness in my local habitat, in my local community. So go into that a little bit more. I'm trying to explain why that's the case. When I have regional processes dominate, that means I have that straight line with a slope less than 1. And this makes sense because what it's saying is basically that as I add more species in the surrounding regions, as I just have more species in the whole entire area that might wander into that local habitat patch, then I end up with more species in that habitat patch. So in other words, the number of species in the habitat patch is not limited by the availability of niches or the availability of sort of roles to play in that habitat. It's actually limited by how many species are out there in the larger area that might potentially colonize the local patch. So again, in that case, more species can fit, and what's limiting the number of species in that habitat, in that local area, is just sort of that availability of what can float in, what can colonize that local area. By contrast, when a curve saturates, when it flattens out, that's an indication that you just can't fit more species into that local region, into that local little community. And that's typically because, or at least we assume that's because, diversity is limited by the availability of space. And it might not be real space. It might be sort of niches, the number of different niches that can fill. And once you've sort of filled up all the niches with the strongest competitor, there's no more sort of space to put more species in. Even though there might be more species that could potentially colonize the area from the surrounding region, they just won't fit. They might land there, but they get outcompeted by everything else that's already filled up the space. So again, that's why we say that local processes dominate when the curve saturates. Now, if we draw a line at sort of that point where the curve plateaus out and flattens and never gets bigger, that's what we call the saturation point, and that's the limit on the local species richness. All right, so that's a little bit about measures of diversity and a little bit about how we compare local and regional richness. So now let's step back again and think about global biogeography. So global patterns in species diversity and composition tend to be influenced by a number of factors. One is geographic area, just sort of how much space there is for things to exist. Another is isolation. So, two regions where I've been sort of isolated from each other for millions of years, maybe they're going to have different patterns in diversity, in, for example, beta diversity, than two regions that have only been separated for a couple thousand years or not separated at all. Another important factor is evolutionary history, sort of what animals happened to be there originally and then radiated to sort of make the rest of the diversity in that area. And then, of course, global climate. We know that things like temperature and precipitation determine who can live where, and so that's a big determinant of global biogeography. In general, at the broad global scale, we recognize 6 distinct biogeographic regions, and these all contain distinct biotas that differ in species composition and diversity. And so these are shown in the map in different colors, and I've got the colors on the map matching the colors that I've labeled them. So that blue area, which is where we live, that's the Nearctic biogeographic region. That's basically North America. The green part of the map, which is Central and South America, that's the Neotropical biogeographic region. The yellow kind of area that encompasses Europe, Asia, and Africa, that is the Palearctic biogeographic region. And then most of Africa, except for that top part, is in the Ethiopian biogeographic region. Finally, we have the Oriental biogeographic region, which is India, China, and Southeast Asia. And then Australasian biogeographic region, which is Australia, the Indo-Pacific, and New Zealand. So those are the 6 biogeographic regions on Earth. And not sort of by chance, each of these regions corresponds to one of the Earth's 6 major tectonic plates. So the tectonic plates and the movement of the tectonic plates, the behavior of the tectonic plates has been sort of fundamental in shaping where continents and land masses have been historically and where they're sort of going to go in the future. And so this, in turn, has been really influential in sort of determining where organisms come from and sort of their evolutionary history and how long they've been isolated and the amount of land mass that they've had to spread out into. So there's really 3 different ways that tectonic plates interact, that things that can happen at the boundary between tectonic plates. The first is what we call mid-ocean ridges. And in this case, molten rock flows out of the seams between the plates and cools, and it basically creates new crust that forces the plates apart. And this forcing of the plates apart is called seafloor spreading. And seafloor spreading is really instrumental in determining how the continents move relative to one another, how those tectonic plates sort of shift and move around through long time scales that are sort of important in determining what land masses have been near each other or far from each other for how long. Another way that tectonic plates can interact is through what we call subduction zones. So in this case, one plate is forced down and another plate is forced up. And this gives rise to areas with earthquakes, volcanic activity, and mountain ranges. So again, this type of interaction is really important for those local features like the mountains and the volcanoes. It's a little less important for sort of the movement, broad-scale movement of the continents. But again, it's important for sort of determining what the landscape looks like where organisms are found. And that in turn governs biodiversity. The final way that tectonic plates can interact is through faults. And this is where 2 plates meet and slide sideways past each other. And again, this can sort of give rise to more landscape scale features of a region. Again, one of the most important sort of tectonic plate interactions, that seafloor spreading, that's what's crucial for determining movement of the plates and thus the continents over long time scales. And so if we actually go back way back in history to the Permian period, 250 million years ago, most of the land mass on the globe was fairly connected. And you can see that in what we call Pangea. And as a result, you didn't have these large ocean boundaries that sort of inhibit dispersal as much. However, if you move to the Cretaceous period, about 100 million years ago, what you see is that seafloor spreading has caused the land mass to split apart and break up into these different regions. And so we can loosely classify them into two different groups. We have Laurasia in brown, and we have Gondwana in green. And so again, now we're starting to separate the land mass. We're starting to create a barrier to dispersal of the organisms, a barrier to spreading of the organisms across those land masses. And that's important both in determining sort of the geographic area that these species have to spread out in, but also in determining their evolutionary relationships relative to one another. By the time we get to the early tertiary period, about 60 million years ago, now you can start to see the world as we know it begin to take shape. You can kind of probably, if you squint, recognize South America, North America, Europe, Africa. But what notice is that, you know, the parts that are connected or were connected 100 million years ago are not necessarily the parts that are connected today. And that's important to understanding why we find certain species in one place relative to another, and where those species originated and then where they've spread to since. So if I take a look, and again, I divide the land mass that is currently sort of spread across the globe into two big groups, the Laurasian group and the Gondwana group, then you can see that sort of historically everything in Gondwana was more connected to each other and everything in Laurasia was more connected to each other. And so really the take-home message from that is that you would expect species to be more similar when you're looking at South America, Africa, India, Antarctica, and Australia, because they all came from Gondwana, which has that shared evolutionary history, that less limitations to dispersal. And then you expect more shared sort of species in the Laurasia group, which is North America, Europe, and Asia. The interesting thing to note is that, again, these are the land masses that have historically been connected. They're not necessarily the land masses that are connected today. And by sort of the same argument, some of the land masses that are connected today, most importantly for us, maybe North America and South America, have only come together very recently. And so that's why the biota differs substantially between South America and North America, even though they're spatially very close together. Now, when these land masses got sort of jammed together, what happened is that we suddenly allowed this corridor for dispersal of species from the previously separated land masses. And so some animals moved up from South America to North America, and other animals moved down from North America to South America. A good example of animals that have moved from North America to South America are the large cats. There's fewer examples of organisms that have moved from South America down to or up to North America, but armadillos are a good example. They evolved and sort of originated in South America, and they have spread up, and now they're in Texas and sort of the southern states. Because the 6 biogeographic regions have sort of been separated for quite a while, they reflect that evolutionary isolation. And so that is, again, why you have these large differences between North and South America, because they haven't separated for a very, very, very long time. They were separated when Laurasia broke off from Gondwana. You have more similarities between Europe and North America because they have that shared history as being part of Laurasia. However, you have much more differences between the organisms you find in North America versus Eurasia than you sort of see differences between sort of the west coast of North America versus the east coast of North America. Again, the longer they've shared a history and the more connected they've been, the more they're going to share that sort of species diversity. They're going to share those similar types of species. All right, so history is really important in determining the patterns we see in biogeography and determining what species are where and how diverse the species are in one place relative to another. But there are other trends that sort of have nothing to do with evolutionary history. And one of the most often observed is the negative relationship between latitude and species diversity. So generally, species diversity decreases towards the poles. And this is true sort of looking from North America to South America, but also looking Europe to Africa. So even these sort of chunks of land that have had very different histories in Gondwana and Laurasia, we still see these same sort of patterns where we see increasing diversity around the equator and less diversity at the poles. Of course, there are exceptions to every rule. That holds true for a lot of different plants and animals, but there are groups of animals where that doesn't hold true. A good example is seabirds. So seabirds are actually most diverse in the northern hemisphere and then second most diverse in the southern hemisphere, and they're not as diverse in the tropics. So very broadly, seabird diversity appears to vary with marine productivity, and marine productivity is actually higher in temperate and polar waters. Another good exception to the rule is salamanders, right? We don't see the most diversity of salamanders around the equator. We actually see it in our backyard. Now, when we have these little pockets of diversity that sort of aren't following the pattern of latitudinal gradients, we actually call them biodiversity hotspots. So we are in what we would call a biodiversity hotspot for salamanders. And these are areas of particularly high species richness for that particular group of organisms. Despite these, fluke trends that are slightly different, for example, seabirds and salamanders, the vast majority of species do follow the trend in latitudinal gradients, where closer to the equators you have more diversity and further from the equators you have less, or at least more richness and less. There's a lot of different processes or a lot of different theories that have been posited to explain latitudinal gradients and species diversity. Regardless of the theory or the mechanism, very broadly, species diversity is governed by three different processes. One is dispersal, which includes immigration and emigration. The second is the rate of extinction of species, and the third is the rate of speciation. So you can imagine I might start with these three salamanders, my species richness is 3, and then if some other different type of salamander immigrates in, now I've got 4 different species, and so I've increased my species richness. Then maybe one species goes extinct, and so now I've decreased my species richness again. But maybe a third species speciates, and now I've increased my species diversity or my species richness again. Now, if I'm looking across local scales, dispersal might matter in terms of governing species richness. But I'm looking at global patterns across the whole globe. It's unlikely that the differences we see on that broad global scale are driven by dispersal. There's no reason to believe that there's sort of different dispersal to the poles versus to the equators. And so generally, most theories that try to explain latitudinal gradients actually focus on the latter two mechanisms or processes that influence diversity, and that is extinction and speciation. This leads us to what we call the species diversification rate. It's basically the net gain in species when you account for speciation and extinction. So you take the number of species that arise because of speciation, that's your speciation rate. You subtract off the number of species you lose because of extinction, that's your extinction rate, and that gives you some net gain or loss in species in a particular area. So, one of the primary theories, or the first theory that's been used to explain differences in species diversity across latitudes, is the idea that species diversification rates differ. Why would that be the case? Why would we have a different net gain or loss in species diversity in the equator versus the poles? Why would extinction and speciation be different? Well, there's a couple differences. First, if I take a look at the equatorial regions, that's where the largest land area is. Just the bulk of the continental landmass is centered in around 26 degrees north and south for whatever reason. It's also the most thermally homogeneous region, right between 25 degrees north and 25 degrees south. We don't have seasonality. We have a very homogeneous, constant area. We don't have as much variation even across longitudes in that area as we do maybe further north. What does that mean? Well, because we have this large land mass, it means we can get large populations just based on the amount of space that can be taken up. And remember, if you think back to lectures previously, we've said that small populations have a higher risk of extinction because just stochastic events, some moose stepping on a flower might cause that flower to go extinct. Whereas if you have a large population, you buffer against those stochastic events. What's the chance that the moose is going to step on all 1,000 of those flowers in that area? So again, just more populations, even if they're in the same sort of physical area, larger size populations are less prone to extinction. And so if I have more land mass and it's thermally homogeneous, I'm going to have larger populations in that area. Also, just the fact that I have more area spreads the risk out over a larger region. And so one little event like a storm that happens in a particular area isn't as bad, isn't as likely to cause an extinction if the animal has a large area to spread out over and there's another part of that area that wasn't hit by a storm. So again, this lowers the risk of extinction. Large populations spread out over large regions, large areas, that spreads your risk, it lowers your risk of extinction. And so the idea is that extinction rates may be lower in the equatorial zone than they are in the pole simply because of this large, sort of thermally homogeneous, all the same land mass. And again, that would lead to higher species diversity right around the equator. Another theory, the second theory, is that there have been differences in species diversification through time. So the equatorial region has been more climatically stable over evolutionary history. By contrast, if I take a look at the poles, they've been constantly disrupted by ice ages. The ice ages, the glaciation, generally doesn't reach the tropics. And so, the northern regions keep having this loss of species to an ice age, whereas the tropics don't. And this can lead to two different possibilities. First, we can think of the tropics as cradles. They might act as the sort of where the species originate. And so you might think of all the species originating in the tropics, and then when you have sort of warmer periods without ice covering everything, these species disperse to the temperate regions. And some of them make it up there, and so we have some species diversity up there from the stragglers that get up, but then they might be wiped out again by the cold. And then they kind of originate in the south and move up, and then they get wiped out by the cold. And so again, you have that lower species diversity up towards the poles because basically your source is the equator, and only some of the things that get invented or speciate at the equator make it up to the pole. So everything is sort of at the equator, but some subset makes it up further. Another possibility is thinking of the tropics as museums. So in this case, you might think, well, the extinction rates, maybe even if speciation is the same in polar regions as it is in the tropics. We don't have higher speciation in the tropics. It's all kind of the same, but maybe I'm losing less of the stuff I had in the tropics, again, because I have that much more stable, climatically stable area around the tropics. I don't have the ice ages. So even if we have a lot of speciation in the poles and at the tropics, maybe the tropics aren't acting as the source, they're not acting as the cradle, but they are the museum because things get killed in the poles by the ice ages and other sort of climatic disruptions, whereas they don't get wiped out, they don't get killed around the equator. The third theory has to do with productivity. And if you remember, we've seen this figure a couple of times where we look at net primary productivity across the globe, and we see that it's pretty high around the equator. During the summer, or during the northern hemisphere summer, it spreads out to sort of our area, but it still remains high along the equator, maybe not in South Carolina, but it still remains high along the equator, and then it's still, again, high along the equator. And so we have this much higher productivity in equatorial regions. And so because terrestrial productivity is higher in the tropics, that leads to, again, differences in risk of extinction. Once again, it's tied to that larger population size. If I've just got more energy coming in, I'm capturing more energy because the sun's hitting it more directly, we got more photosynthesis going on, then that allows for larger populations. And again, larger populations lower extinction risk, just because you're less likely to lose a population due to a chance event. They also allow for greater species coexistence. So the more energy I have to go around, the more likely I'm able to sort of partition that energy into little groups or little niches where I have multiple different species living together. All right, so that's sort of global patterns in biogeography. If I look at regional patterns in biogeography, they are not necessarily influenced by the same factors. In fact, they're often influenced by things like area and distance. And in fact, often regional biodiversity is determined by the balance between immigration and extinction, rather than that balance between speciation and extinction that we see on the global scale. So this leads us to sort of trying to understand how biodiversity changes as a function of area. And one way we can understand that is to plot what we call a species-area relationship. So this is a relationship between species richness and the amount of area you're considering. And typically we do this by sort of counting the number of species in one little area and then expanding our view bigger and bigger to more larger and larger regions and figuring out how many more species we gain. And so this is just a plot from an area in England. Sometimes it's hard to figure out where to draw the line to plot these plots of species richness versus area in sort of a very generic landscape, say in South Carolina. So instead, scientists often use islands. They often study this increase in species richness or increase in the number of species as a function of area using islands where it's very clear sort of different islands have different areas and you can look at diversity. This works well for mainlands as well because sometimes on the mainlands you actually have things that function as islands. So I can think of sort of different lakes and I can look at the number of species in these different lakes as a function of lake size. That's sort of the same thing except my islands are actually little lakes embedded in a terrestrial environment. I can also think of mountain islands. In this case, it's sort of, if you think about the Smokies, there are those high elevation spruce fir forests at the top of the peaks. And, you know, at each mountaintop you might, or at least at the high mountains, tops of the high mountains, you have these spruce fir forests. They're kind of isolated from each other, but they form this connection of little patches of spruce fir forest as you go up the ridgeline of the southern Appalachians. And so again, they act like islands. We can also think about habitat fragments. This is commonly done in the prairies where we have natural prairie embedded in farmland, and we can look at, well, what's our species richness in our little patches of natural prairie as a function of the patch size. How many more species do we get in a patch when we make or when we preserve larger patches of natural prairie rather than turning them into cornfields? So again, when I look at species area relationships, one important feature is the slope of the curve, which reflects how fast diversity increases with area. So how much more of a gain in species do I get as I preserve or as I make a region with a larger chunk of habitat? So when I have a very low slope, like that sort of dotted line with the plus signs, that indicates that you need a large change in area to see any kind of change in diversity. So there, you know, if you want to preserve a lot more species, you got to preserve a lot more area. If I have a much steeper slope, that indicates that even a small change in area of a particular habitat fragment or habitat patch or island is enough to see a huge change in diversity. And so that's an area where just preserving a little bit more habitat may be really important to keeping species. It's not just area, though, that matters to regional scale diversity. Many studies have shown that island size alone doesn't explain diversity. A big feature is distance to the mainland source. And so islands that are more distant from a large landmass or a larger island or a much sort of the mainland source population tend to have fewer species, and islands that are really close, even if they're small themselves, if they're close to a really big landmass or a really big island or a really big area that has the species that they also can sort of provide habitat for, then they're going to have more species. And this ultimately led to the theory of island biogeography. And it was developed by Robert MacArthur and E.O. Wilson. And generally I don't make you learn names, but in this case I am going to. So a couple of reasons. So first of all, MacArthur spent some time in Toronto, which is my hometown, and he was a mathematician. And I love math biology. E.O. Wilson studied ants. And so the theory of island biogeography came out of Canada, math, and ants. And that's why I want you to know these two people. They're actually some of the most famous ecologists in the world. And this is one of the primary theories in all of ecology. Basically, the theory of island biogeography was based on observations and maths. And they found that a 10-fold increase in island size caused a doubling in ant species diversity. And then they also tried to explain this using math based on sort of this idea that the number of ants on an island, or the number of anything on an island, is really this equilibrium process between species gain and species loss. So to look at that in a little bit more detail, we have this very famous kind of plot. And there's sort of a couple of assumptions that go into the theory of island biogeography. The first is that immigration rate, or at least the rate at which immigration leads to a new species on an island, decreases with the number of species already present on the island. And so you can see that. Here I have my mainland source with all the birds, and on my island. And if I have no birds on that island whatsoever, every immigration event leads to an increase in species richness. So I had 0 for my species richness before. When I have an immigration event, say a crow, I now have 1. Now my next immigration event is also likely to increase species richness. It was a cardinal. And maybe a pigeon arrives, and now I've got another increase in species richness. But now I've got quite a few birds on this island. I've already got some species richness going on. And so maybe my next immigration event doesn't change anything. And maybe it takes a whole pile of more immigration events before I see an increase in species richness. There, I finally got one. And so again, it's that idea that sort of the rate at which immigration leads to an increase in species richness decreases as you get more and more species already built up on the island. The second assumption or hypothesis that goes into the theory of island biogeography is that extinction rate actually increases with increasing species present. And the idea here is that if I've got a lot of species on that island, some of them are probably going to be rare and some of them are going to be abundant. And if I just randomly shoot one of them, and I've got a whole lot on there, then at some point I'm likely to hit some of those rarer species and they're just going to go extinct, right? However, if I don't have too many species present on the island, most of those species are going to be abundant. They're the only things there. And so the chance of me shooting them and wiping out a whole entire species is much lower. So again, that's that idea where as I have more and more species present, any random chance sort of death event is more likely to lead to some small population going extinct. Whereas if I've only got one species present, it's taking up the whole island with a single species, then if I kill one bird, it's not going to lead to extinction. So those are the two general curves. The rate at which immigration leads to a increase in species richness decreases as I get more species present, and the rate at which extinction leads to a loss of species richness increases as I get more species present. But that's not the only story. The next assumption that goes in is that all else being equal, small islands should have higher extinction rates than large islands. And the idea here is that small islands, even if sort of the distribution of animals over both islands is the same, small islands just have smaller populations. And so a bad event, maybe a moose stepping on a flower, can have a much more likely chance of wiping out that particular population, of causing extinction, because that population was just that much smaller, initially. So if I have sort of one big island and one small island that's half the size, if they have the same number of species on it, then generally we can expect that the small island that's half the size would have sort of half the size of populations as well, and so again, much more likely to have extinction events, and that skews it so that you don't end up having the same number of species on both islands. Sorry, and then again, what you'll notice there is that extinction rate, in general, it increases with the number of species present, but for large islands, that extinction rate is always lower than it is for small islands. The next and final, fourth assumption that goes into the theory of island biogeography is that all else being equal, islands that are near to the mainland should have higher immigration rates than islands that are further away. The idea here is that, whatever, birds, insects, seeds are just more likely to survive if they're blown from the mainland to some very close island. And they're just less likely to get across the ocean if they're trying to get far out to some very isolated island. And so you just have more flux of immigrants into close islands than you do into far islands. And so as a result, even if each immigration event was equally likely to cause an increase in species richness, you just have more immigration events to those nearby islands. And so again, immigration leading to an increase in species richness decreases with more species, but it's different for islands that are close to the mainland versus far. We see that that red line is much lower for islands that are far from the mainland and much higher for islands that are close. And again, that's because that further distance makes successful immigration less likely. So ultimately what this does is it allows us to figure out where that balance, where that equilibrium between species gain and species loss falls, and how that depends on where the island is and how big the island is. So this allows us to sort of try to predict the number of species that might be present, at least relatively. If I take a look, for instance, at a small island, something like that one that I've got in the bold black, that's located far away from the mainland, then I'm going to have a smaller number of species than if I take a look at some large island that's located really close to the mainland. And so by doing that, we can sort of understand how the number of species, or how the species richness on different islands varies as a function of their size and also their distance from some larger source population. So that's our lecture for today. I hope you enjoyed it, and if you have any questions at all, I'd encourage you to email me. Otherwise, I will see you next class.